All right. So, hey everybody, I'm Derek Kraft. I'm a third-year PhD student at the Hawai'i Institute of Marine Biology. Thanks for showing up today on this lovely sunny Hawaiian day. So I have the privilege of working with Richard Coleman, who showed up today, and Dr. Brian Bowen. And we have a project we call Fish Flow, and it talks about how far fish would go to get away from their parents. So, the lovely Hanauma Bay, I'm guessing maybe none of you have been in it today, but who's been in it at some other time? Yeah, what do you guys see when you go in the bay? Fish, good. Glad somebody said fish. It's a fish talk. Yeah, so there's lots of fish out there. And even though this bay has been protected since 1967, and it's this beautiful, thriving ecosystem, this bay very much depends on other parts of the ocean for its sustainability and its liveliness. And just the same, other parts of the ocean rely on this thriving ecosystem to keep it sustained and to replenish its populations. So that's really important, and something that's a big driver of that is the life cycle of reef fish themselves. So they start out as adults down here, and reef fish are broadcast spawners. So they're going to shoot their gametes out into the ocean, and they're going to fertilize and then turn into larvae. But this whole top half of their life cycle is out in the ocean and away from the shore, and it can last anywhere from a couple of weeks to a couple of months. And during that time, we're not really sure how far they're traveling or where they're going. Sorry about that. Then the larvae settle and become recruits on the reef and grow from juveniles to adults. In this bottom half of their life cycle, they have pretty small home ranges, so they're not moving super long distances. So those connections are really important. They're important for managers. And here's an example of that connectivity. Mark Christie and his team, they looked at the yellow tangs over on Big Island, which is a really important species in the aquarium fish trade. And over time, the fish trade wasn't doing so well over there. And so they set up these marine reserves to help replenish that trade. And it worked. The aquarium species started coming back, but they weren't exactly sure why it was working or how. So what they did is they looked at the genetics of the adults here. Hi guys, welcome, come on in. So they looked at the genetics of the adults and compared them to the genetics of the recruits that they found, kind of like a DNA analysis you would do trying to figure out who the parent is of an illegitimate child. So similar to that. So what they found was they found some connectivity. They found that down in the southern part of the island here were directly linked to some of the juveniles they found up the coast. And that's really important, especially when thinking about these marine reserves and how they're connected. And if they're replenishing each other, or if they're just replenishing themselves. So they got to see that the southern parts was important to the rest of the island because it was seeding the juveniles up the coast. And that was correlated with the currents running up that coast as well. So the fish are important for the aquarium trade, but they're also really important for people to eat. So historically, reef fish has contributed a lot of subsistence, and people go out and fish these fish and they eat them, they feed them to their neighbors, they feed them to their families, and currently about 31% of Hawaiian residents still go fishing recreationally, which includes to eat. And fish are also important, like why some of you, well, maybe didn't come today, but on some days come back to the bay to see the fish as far as ecotourism. Unfortunately, the reefs are changing a little bit. So those recreational fisheries still count for about 36% of harvest here in Hawaii and also in other southern Pacific islands. And that fishing is pretty unregulated. So it's that high fishing pressure along with invasive species and coastal development has shown a real fundamental shift in these coastal ecosystems. And these target species, these nice reef fish, are a little bit in decline. And that raises some concern. So the Hawaiian community leaders got together and they created a list of species that they were concerned with. And we took a look at that list and we narrowed it down to about these 5 that we thought we could realistically get enough samples for and look at. So they are the square spotted goatfish, the manini, also known as the convict tang, kole, the gold ring sturgeon fish, mullet down here, and then also the amilu, which is a bluefin trevally. And these are all really important for local fisheries and for people to eat. So what we really need to do is protect the important areas, the areas that they spawn, the areas that recruit. And to do that, we need to know where fish are coming from and where they are going. And that's what we plan to do. So what we would like to do is link the locations where these adults are spawning to where the recruits are settling. We're going to do that with these important species, and we're going to do it across all of the main Hawaiian Islands and mostly track how far fish are going away from their parents. How we do that, we collect samples of reef fish, we collect adults, and we also collect juveniles, non-lethally when we can. We extract their DNA and put it through a high-throughput sequencer, and then we get that information back in a computer, and that's where we do our little parentage analysis and compare the adults to the juveniles. And by linking adults to juveniles, we can see how far they've traveled. Where we do that. Okay, so this is a map of Kane'ohe Bay. So we're at Hanauma Bay, which is about here, and Kane'ohe Bay is basically up around the corner. All these little black dots represent little patch reefs where we've sampled adults and juveniles. So this is looking at connectivity on a very, very small and tight scale in this relatively predictable body of water. So this map over here is showing residence time of the water. So the blue up in the northern part of the bay, this water is exchanged in and out of the bay, around between 1 and 5 days. All of that water is exchanged in and out. We're down in the southern part of the bay, the residence time is much longer, 25 to 30 days. So this water is not moving nearly as quickly. And okay, let's look at a little model. This is a model put together by one of our colleagues. And what this is doing is showing water movement within the bay. So let's pretend all the, oh yeah, I gotta click it over here, sorry about that. So you're gonna see these little dots, and let's pretend these little dots are fertilized eggs, broadcast spawned into the bay. And we have different colors, the red and the yellow, and also the green and the blue here. So pretend those are all different genetically. So those are different eggs and turning into little larvae. And this is how they move about the bay, just as the water movement. Now notice what happens to the yellow and the red ones. They start to move towards the mouth of the bay, and then whoop, they are pushed out. And pushed down the coast somewhere. We're not really sure how far they would go. But notice that the green and the yellow ones, or the green and the blue ones, sorry, have kind of stayed in the bay. So what this could possibly suggest is that fish down in the southern part of the bay are seeding the rest of the bay, or replenishing the rest of the bay, and fish that are up in the northern part are moving on elsewhere. So again, Kane'ohe Bay here is one small piece of the puzzle, even though it's kind of the center of the bullseye for this study. So we saw those eggs leave the bay. How far are they going? Are they going up around the coast? Are they going all the way to the north shore, maybe down to the west side? We're not really sure. And also on that same note, are eggs and larvae from Kailua and Waimanalo, are they going in to seed the bay? So that's kind of what we're looking at as we scale back here. So we're going to also sample fish all around Oahu and see how they're connected between different sides of the island itself. But now, just as Kane'ohe Bay is a small part of Oahu, Oahu is a small part of the big Hawaiian archipelagos altogether. So we're also going to be collecting fish from the big island of Hawaii, Maui, Kauai, Niihau, and seeing if there's connectivity between the other islands as well. So what we expect to find, we have no idea, right? So that Kane'ohe Bay was kind of a predictable little body of water. When you start getting into bodies of water that consist of an entire island or an entire island change, it becomes much more unpredictable. So we're not really sure, which is also why this is important. We're excited to do this study. There are a few things, though, we can maybe predict. These large expanses between Big Island and Maui here, these dotted lines, and between Kauai and Oahu, several marine species have shown genetic breaks between these. Also, because they're a large distance, there are really deep water currents that run through there, so that can prevent those little larvae from getting across the islands. And we can also make a pretty good assumption that these little larvae and these eggs are going to flow with currents. So we have the North Hawaiian Ridge current here and the Hawaiian Lee current here, all of them kind of pushing to the west. So from Big Island pushing through to Kauai. So we kind of expect to see some patterns of distribution that direction. Importance of these results. So this is going to identify critical areas of spawning, critical areas of recruitment, and these are all really vulnerable to overfishing and need to be properly managed. So these results can help local managers and state managers when they're doing things like designing restricted fishing areas that'll help protect these species of interest. Kind of like these, so this is a good example of already created regulated fishing areas. And some larger impacts, it'll help us really understand how neighboring islands influence the populations here on Oahu and how Oahu influences populations on other islands. So the main theme of this talk is we are looking at the connectivity, not just on a small scale, but on a large scale across the whole Hawaiian archipelago. So the next time you're swimming here in Hanauma Bay or maybe some other place you enjoy snorkeling or hold dear to your heart, you see a cute little reef fish, you can think to yourself, where are your parents and how far have you gone running away from them? With any large-scale study like this one, I have so many people to thank who've come out and helped us fish. In other parts of the research, especially our advisor, Dr. Brian Bowen, who is also awesome enough to be here today, and Richard, who's working on this for his dissertation. He's really the brains behind this whole operation. And not to mention, we have lab mates, we have people from other labs who come out and help fish, and even some citizen scientists who showed up today, which is nice of them. So thanks to all of these lovely people and to the Tobo Lab as a whole. Also for Hanauma Bay for letting us come out and present with you guys today, and Sea Grant for their continuing efforts, doing great things, not just here in Hawaii, but around the world. So thank you, and I'll take questions later, not now.
Hi everybody, my name is Lindsay and I am also a member of the Tobo Lab. The toe is not here, but the bow is. Thanks for coming, Brian. So I am a fourth year PhD student, and today I'm going to be telling you guys about what I'm doing for my second chapter, which can be summarized by the title Habitat Suitability Modeling in Penguin Bay in Molokai.
Excuse me, where is Penguin Bay? Never heard of it.
I am about to show you. Hang tight. All right, so I'm structuring this talk with the where, the what, and the how. So the where is what you just asked. So where is our study site? What exactly is habitat suitability modeling, and how do we build these models? So where is Penguin Bank, Molokai? So Penguin Bank is a submerged shield volcano off the southeastern coast of Molokai. And it's about 1,000 square kilometers. Its depth range is something like 40 to 200 meters across the entire plateau. And on the right side of the slide, you can see some different pictures I've taken when I was reviewing the sub-dive footage. So on the top here, we have some calcareous algae blowing in the currents. In the middle picture, we have some plate corals over here in this corner and some more halimeda and just different types of substrate. And then on the bottom, you don't see much at all. And that's just a lot of turbid water to demonstrate that the currents are really ripping across Penguin Bank. All right, so the other part of the where question. So my zone of interest, the oceanic zone that I'm studying, is the mesophotic zone. So the depth range of this mesophotic zone is between about 100 to 600 feet or 30 to 180 meters. Gives it away in terms of its light profiles. So mesophotic means mid-light or low light. Here we see about 10 to less than 1% of surface solar radiation reaching this zone. As for temperatures, we see temperatures pretty similar to what we see at the sea surface around the upper mesophotic, but then you lose about 25% of that heat as you travel down towards the lower mesophotic. And in Hawaii, that would translate to about 60 degrees Fahrenheit. We see decreasing currents as depth increases, and we also see specially adapted organisms like corals, fishes, and algae. And when I say specially adapted, I mean they are particularly well suited to living in this low light and cool environment. So how does one get to the mesophotic zone? Well, it's not easy. So if you or I wanted to travel to the mesophotic, it would be pretty hard. It's the upper depth limit at about 30 to 40 meters is around the lower depth limit that a recreational diver can safely access. So scuba diving is usually out of the question unless you're using really specialized equipment or special blends of gases. So you might say, okay, why don't you take a submersible down there? Well, submersibles cost a lot of money to operate, usually about $10,000 to $20,000 a day to operate. That's a lot of money, and when you're spending that kind of money, you want the most bang for your buck, and that usually means surveying something like the Marianas Trench, which is about 11,000 meters in depth, not 100 meters in depth. So it's pretty hard to survey, and this is where habitat suitability modeling comes into play. So habitat suitability modeling can be explained really as a combination of two types of data. Observational data, so that camera or video footage that I showed you guys at the beginning of the talk, pictures of fishes or corals or algae or any other invertebrates, anything we might be interested in, or pictures of nothing. That's data too. We want to know where organisms are and where they are not. And we combine that observational data with environmental data. So this is data that pertains to the abiotic components of your study region of interest. This can include temperature profiles, light profiles, maybe information about the character of the seafloor, waves or currents, and so on and so forth. So combining those two types of data looks something like this. We have a study area and we have these little points all across the study area. And each of these points is an observation of whether your animal or organism of interest is either present or absent. And then you can match that point using coordinate data with corresponding environmental information. So how would we build one of these habitat suitability models? Well, first we want to consider our hypothesis or our goal. So you might want to consider, are we interested in the distribution of a sessile species like some type of coral or sponge? For me, I'm interested in the distribution of Leptocerus, that's this plate coral in this lower picture here, or maybe a mobile or migratory species like a salmon. We might want to model organisms across trophic levels. We might want to say, okay, I have a model that I want to set up for different families of corals, or we might be interested in one species of fish. Next, you have to collect your data. So these are the environmental and observational data that I was referencing. So that can be sourced from satellites or buoys, from sub footage, from research cruises, or even other models. Next, you have to set your resolution. And in large part, this will be determined by that environmental data that you collect. Some of it is available at pretty low resolution, meaning that it's very big resolution. So you don't have a very fine scale. So you might be restricted to modeling in square kilometers, or you might be able to downsize that to hundreds of meters, or even better, you can go tens of meters or finer. I am modeling at 5 by 5 meters squared, so that's pretty small. Next, you want to build your model, and there are many, many ways to do this. Essentially, you are going to put together an equation where you determine the probability of your organism being present based on different values of your environmental variables at a particular point times the effect of those variables at that particular point. And you can modify the effects preliminarily based on clustering of your data points. That's something that we want our model to know about and to take into account. Or we can say, for example, we have this observation I in a particular pixel or at a particular point. We know that mathematically it's easier to break up a map into these pixels or these different points, but in reality, a habitat is continuous. We don't actually go out snorkeling and see this grid of different organisms. So we can say, okay, I might have this observation I in this particular point, but I also want to incorporate environmental information from surrounding pixels. So you can do that too. I happen to be doing all of these and another technique as well, and just averaging the results, and it's called an ensemble model. So after you've built your model and run it, you want to interpret your results. So what you're doing here is essentially looking at probability distributions produced by your model. So on the y-axis, we have a number of corals observed at some value of your environmental variable. Here, let's just say it's depth. So for me, I have certain corals at this low depth range, certain corals observed at this high depth range, but then I have a lot that are observed at this particular range right here. So I can say if I have some sort of observation at some depth value that is at this star, so near the peak of this depth range, that's probably a really suitable habitat for those corals, because we see a lot of them colonizing areas at this particular middle depth near the peak of that bell curve. And so to translate this into just an English sentence, we can say that the probability of Leptocerus presence depends on a combination of environmental factors, including depth and distance from shore, hardness of the seafloor, the complexity of the seafloor, and the slope of the seafloor. And that is actually true. Those are the results of my models. So you guys are the first to hear that. And you have to share your research. So that's what we're doing here today. You can do that by just telling people about it using the sentence that I just shared with you. But even better, you can create these aesthetically appealing maps. So I created a couple maps and I just put some up here for you guys. So the orange and the red colors indicate areas of high habitat suitability for my organism of interest. And the yellow or the blue areas indicate areas of low habitat suitability. So one question that I have not talked about so far is why. So why am I going through all this trouble to build these models? Well, as we know, climate change is the greatest threat to our species, and it's not so great for coral reefs either. It's killing many of them, and the projections are not good through the next several decades. So we see a lot of coral bleaching going on. We also see a lot of coastal development as human populations increase around the world. We see that humans are interested in advancing technologies that will help our species, such as wind turbines off the coast or laying seafloor cabling. And all of these events and actions have impacts on our coral reefs. Now, we are interested in mesophotic reefs because they can be considered a potential continuation of shallow reef systems. The mesophotic corals generally are thought to maybe reseed these shallow reefs by spreading their gametes or their baby corals up to the shallows that have been degraded by the effects of climate change or coastal development. And mesophotic reefs are also thought to potentially be a nursery for juvenile fish. So we don't know these things, but when you want to know something about a species or a group of organisms, you better know where they are. That's the first place to start. And so that's what I'm doing by creating these maps. I'm letting resource managers know where they are, scientists, and hopefully we can respond appropriately to these threats that face us and use these maps for good. So with that, thank you for listening. I want to thank all of my data providers, too numerous to name here, everyone on my committee and everyone who's helped me with this project to date, and particularly the Hawaii Undersea Research Lab, which has cataloged all of that submersible data that I have been using. And with that, hand it over to Leilei.
Hi there, I'm Leilei Shih, and I'm a 6th year PhD candidate in the Oceanography Department at UH Manoa. And today I'm going to be talking about an invasive sponge called Mycali grandis. It's a bright orange sponge that is prevalent in Kane'ohe Bay and some other shallow water ecosystems around the main Hawaiian Islands, and specifically about the ecological impacts of this sponge. And my advisor is Brian Popp, who is in the Oceanography and Geology and Geophysics Department at UH Manoa as well. So a little bit about sponges in general. Sponges are actually a marine animal, not a plant, and they arose some 600 million years ago during the Adiacardian period, which was before the famous Cambrian explosion, which gave us a rapid rise in diversity in multicellular life. In fact, sponges were the first multicellular organism on the tree of life. Before that, they were just mostly single-celled organisms or assimilations of identical cells. And during the Paleozoic era, which was several hundred millions of years ago, they comprised probably more than half of the biomass on reefs. So they were have always been a very important component of marine ecosystems. In addition, sponges have survived several mass extinctions. Probably the most commonly known one is the one that killed dinosaurs, and they survived that one as well, but also some bigger extinctions. And they are key ecosystem player in aquatic habitats. And this is due largely to their ability to actively pump enormous amounts of seawater through their bodies, which they do to to filter feed, and to also remove waste. And I'll be talking about their pumping ability in a little bit. And also, their body plans are filled with really robust microbial communities, and that's the other component of why sponges play such a key role in ecosystems. I'll be talking about that in some later slides as well. So a little bit about the sponge that I want to talk about today, though, Mycale grandis. Mycale grandis is an invasive species in Hawaii. It was first spotted in around 1996, and it lives in shallow coral reef ecosystems around the main Hawaiian islands, such as in Kane'ohe Bay on the windward side of Oahu, in Kihi Lagoon by the airport, also in places like Lahaina Harbor on Maui. It came to Hawaii probably as biofouling on ships, either on the hulls of ships or in the ballast water. And it originated from the Indonesian, Australia, Malaysia region of the Pacific. We can assume that the sponge is a very resilient animal because it survived a journey all the way across the Pacific, and it was able to establish itself in a brand new ecosystem. Now I just want to take this moment to reinforce that just because a species is not native or introduced doesn't necessarily mean it's bad. Some species such as lilikoi and plumeria were also introduced, and it's good that we have them here. But what distinguishes bad or invasive species is that it can compete with endemic or native species, sometimes causing their extinction. And in that sense, some non-native species might be unwelcome. But what we do know about Mycali, fortunately, is that its population in Hawaii appears to have stabilized. It's no longer taking over, if you will, but it does compete for coral, for space where it does exist. And as I mentioned before, it can have a big impact on the chemistry of water because of its microbial biogeochemical activity, which I will get to also. So just a quick bit about invasive species and why this is important in Hawaii before we continue. Hawaii is one of the most, oops, sorry about that. How do you go back? So Hawaii is one of the most geographically isolated places in the world. And what this leads to is a high rate of endemism, or what that means is species that are found nowhere else geographically in the world. And also these species tend to have very specific adaptability, meaning that they're used to very certain environmental conditions. They might just have a very specific food or a very specific environment, versus non-native species, which have come from somewhere else. They're able to adapt to a variety of environments. In fact, how unique Hawaii is the reason why it's sometimes referred to as the endangered species capital of the world, because we have so many endemic species here. Almost a third of endangered species in Hawaii, or in the country, are actually located here in Hawaii. So it is a very big issue here. As I mentioned, invasive species are highly adaptable to new environments, but also environmental changes. So if we're changing the environment due to climate change, or due to human activity, or due to runoff, invasive species will better be able to adapt, whereas typically endemic species won't. And also, non-native species can compete with endemic species in very specific ways, either for food, or in the case of coral and sponges, sponges can actually compete for space with a coral. So wherever a sponge grows, a coral can no longer grow. So one of the reasons why sponges are so play such a key role in their ecosystems is that they have these really tremendous rates of seawater pumping. Again, they do this, they actively pump so much seawater because they do it to filter feed for food. That's how they feed, but also to get rid of waste, such as ammonia. So they're trying to pump their waste products away from them. So actually, sponges are able to pump so much seawater that they're pumping 50,000 to 100,000 times the volume of their own bodies every single day. In other words, one sponge has the ability to overturn an Olympic-sized pool's amount of water in just one day alone. So that's a lot of seawater that's coming through the body plan of a sponge, and thereby being influenced by the sponge animal itself. So here's an illustration of a simple sponge, and basically the sponge is pumping in seawater along its periphery, along the outside of its body, through these pores, through pores all over its body, tiny pores called ostia from all directions. And the water is actually being pumped by highly specialized cells called choanocytes that have these flagella, or a long tail that spin kind of like a turbine, and they're using this energy to pump seawater through it. And then the water goes through the body of the sponge, it takes out what it wants, it expels what it doesn't need, and it's pumped back out through its osculum. So sponges have either a few oscula, just a few on its body, or it will have one large osculum. Even though water is taken up from all over, it'll just come out in one place. So these blue arrows in this picture are showing the movement of water in the way that the sponge is pumping it. So here is a video of, here we go. This is a video I took of Mycali grandis pumping fluorescein dye. This is a sponge that I collected in Kane'ohe Bay. So fluorescein dye, first of all, it's just a dye that is made from water and alcohol. It's often used on life rafts during shipwrecks to make them more visible in the ocean, and it's also used as a food additive. So it's not as scary as it looks or sounds. But here you can see the sponge is actively pumping dye along here. This is one of the main jets that it's pumping out. So what I did was I injected fluorescein dye all along the outside of the sponge. It's being kind of taken up all along here. It's going into the sponge, and then it's being shot out in these streams. There's one here, which is probably the most visible one. There's another one here, and there's some back here. But you can see from the ripples, you can see some rippling in this jet here. You can actually see how actively and quickly Mycali is pumping seawater at all times. So again, this high seawater pumping rate, coupled in tandem with its very active microbial community, is the reason why it has such a strong impact on its ecology. So sponges actually host a very, very rich microbiome, a community of archaea and bacteria within their bodies. And when the seawater is pumped by the sponge through its body, it's actually exposed to all these microbes in the sponge. So sponges are often called microbial fermenters. They're kind of like, you can sort of think of how we have microbes in our stomach that help us digest and actually do a whole host of really important things. So they are exceptionally rich in microbes that are actively performing biogeochemical reactions at all times. In fact, 1 cubic centimeter of sponge can host over 10 billion microbes in it. So this size piece of sponge can have 10 billion microbes in it. This is up to 1,000 to 10,000 times more concentrated than the microbial concentration in seawater alone. In fact, from microscope studies, we've seen that up to 35% of the space in sponge tissue, over a third of the sponge, is being occupied by microbes. So it's a very significant part of the sponge. In fact, a lot of time when we're talking about sponges, we're not just talking about the sponge itself, but of course the entire microbiome and what it's doing. So what these microbes are doing is they're actually actively changing the chemistry of the seawater around them. The microbes are actually taking up some chemicals that are in the ocean, such as ammonia, and they can convert it into other chemical components, such as nitrate. So here's a sort of an illustration. So we're having ammonium, which is a form of nitrogen in the ocean, being uptaken by the sponge, and the bacterial community converts it into something such as nitrate, which is something that we know Mycali does do. So why this is important, anybody who's ever tried to grow a garden knows just how important nitrate levels can be. And since you're a patron of Hanauma Bay, you've probably heard of how important nutrients can be on coral reefs. If you have too many nutrients in the water, usually from runoff, you know that nutrients can harm coral by causing algal blooms and causing algae overgrowth on coral. So this is something that's very important since the sponges are growing on coral reefs. So learning about the impact of Mycali grandis on coral reef ecosystems requires looking at it from many different directions. Actually, very little is known about was previously known about Mycali grandis. It hadn't really been studied in the past. It's just always been around in Indonesia, Malaysia, and Australia. They didn't really have concern. And as I mentioned, it's fairly new in Hawaii, so we don't know much about it yet. But to learn more about it, we do a variety of experiments on it, such as we're identifying its microbial community through genetics. I'm doing nutrient incubations to determine what it's doing with nitrogen cycling in the ocean. This is just where sponges that I'm studying are being kept in Kane'ohe Bay at Coconut Island at the Hawaii Institute for Marine Biology. This is thin slicing of the sponge. We're looking at it, at the structure of the sponge under high-powered microscopes, studying bulk isotopic composition of the sponges that tells us what exactly the sponges are eating and what they're uptaking. And this is just studying of Mycali grandis in general. And this here is Hawaii Institute of Marine Biology, which is located in Kane'ohe Bay, just around the corner, where a lot of this research takes place. Thank you. Alright, so we'll take some questions.
It's not a question, but I don't know who did it, but people who know me know that I'm a stickler for correct Hawaiian pronunciation. And I was so happy to see that you had all the diacritical marks on all the place names. That was wonderful. Who did that? You did that? Wow. That's good to see.
Thanks.
I think my question goes to Lindsay. Lindsay, you build a model, right?
Yes.
And in the model, I think, from what I gathered, you have some variables. And I gathered also from reading the screen, that one variable is called hardness,
Yes.
and then another one, slope and complexity. Could you define and tell us what hardness, slope, and complexity
Yes.
actually
So
refers to?
the latter part of all of those terms was of the seafloor. So when I say hardness of the seafloor, I mean, is it a pile of rocks that we're looking at, which is really great for corals to colonize because they need hard substrate to hang on to, or are we looking at a sand flat where it's not actually possible for a coral to colonize unless there are little pebbles that maybe they can latch onto? And then for slope, are we looking at a flat plateau, or are we looking at a cliff that's more like this, where they're gonna maybe be in a ravine? And then complexity, again, are we looking at a flat surface like this floor, not very complex, which is great because we don't want to trip when we're walking, or are we looking at something like a bed of gravel that has a lot more divots in it, maybe stuff can colonize in those little divots?
Let me ask you a question. There's one slope that is 34 degrees.
Yes.
Now, and you're gonna make your observations for the coral or whatever you're looking at. And when you get the observation, you translate it into a probability? Is that what you do?
Yeah, so that's an easy way to explain it. So essentially what the models will do is tell me what is the probability that this organism is going to be present at this range of values for, let's say, slope.
How do you determine that?
Well, it's like an equation where I wish I could write the equation for you to explain it, but essentially that equation that I had up there, which was probability of presence for Leptocerus coral, which is my coral for my study, equals, then you have effect of a certain variable and the value of that variable. So the value for what you saw up there, which was slope equals 34, that would be 34 degrees. And is actually determined by the model. It's a process called logistic regression. And I could
What kind of regression?
Logistic regression. Yeah.
Oh, go ahead, Steve. This is a question for Leilei. Are you or anyone else doing any studies of the lemons with Molokai and the lemons? Because I did it in 2003.
Yes. Yes, so I did some just a handful of preliminary transects last year, and I would say compared to, are you Steve?
Yeah.
So I compared to your studies, which showed somewhere between like 6 and 12% cover, it looks way below that. But I'm actually, yeah, once the winds and rains die down, I'm actually going back out with my advisor. I could use his help to kind of get some better transects, and then I think we can compare to your data. To do it as comprehensively as you will, because you kind of did the entire bay. Well, I'll be focusing on the South Bay. Yeah, exactly.
But if
So
that is indeed the case, do you have any explanation? How does this happen?
my theory would just be that it's sort of the new baseline. This is just a new level of stability. I don't have any reason otherwise. I think it's just kind of reached a stability. I don't think it's declining. I don't think it'll just magically disappear on its own, but it's not getting worse as you might have feared. I suppose you mean ammonia in the bay. Ammonia in Kane'ohe Bay is sky high, so I think it has plenty of. But that's a great question, and one I should think about.
Yeah, Brian.
Oh, thank you. Great presentations. This is for Leilei.
Mhm.
What eats the colony? Anything?
So I even emailed him a couple months ago this exact question. I don't think anybody knows if anything specifically eats Mycali. So sometimes turtles will eat them some type of way at some sponges. Mycali's kind of a tougher. It's not as soft as some sponges. It's definitely very spicularly when I have handled fragments of it. So I don't know anybody personally who has observed anything eating Mycali. I know that Steve Cole's group was exploring the usage of blasting them with oxygen. That might be one way to air to control their spread, but I don't think they have any. I don't know of any natural predators.
So you are going to be looking at recruitment across all of Hawaiian Islands. How often are you going to be able to go and sample those different patients? Get influence from seasonality.
Yeah, absolutely. We are going to go and try to do most of our outer island stuff early this next year, right? Richard? Good, yeah. So we'll be doing most of it January, February, hopefully. And yeah, you're right. So if, let's say we went to the Big Island and sampled a bunch of fish, this year, 2 years to go to Kauai and sample. I mean, the thing with these projects is they're a snapshot. They're a picture of right now what is happening. So yes,
We're
but
gonna do all the samples at one,
same
in
season
January, all right.
as tightly as we can. Because yeah, it's gonna.
During other seasons? Like one shot.
Most of the time, it's just kind of a single snapshot. I mean, if we could get funding and continuously sample all the other islands in the world, so yeah, I'd love to. Not sure. Yeah. Yeah, right. But yeah, that's a good question.
I have a question for you.
Mm-hmm.
Haley, first of all, I was fascinated about sponges because I don't know anything about sponges, and I was really amazed at what they can do. Now, do you know, or does somebody have a theory of why they pump so much seawater? I know we got microbes in there a lot,
Right.
but 50,000 to 100,000.
It's a lot, right?
Yeah. What's the reason for so much pumping?
I would say that there are a multitude of reasons. So kind of what was always traditionally thought is that they're filter feeding. They literally feed, they filter out tiny particles from the water to feed themselves. But also because they have these microbes in their body plan, which you can kind of say that they're farming these microbes. These microbes can provide the sponge with nutrients it needs, with energy that it needs. It's actually also feeding its microbial community. So that's kind of the traditional thought of it. And also remember, sponges are a very prolific organism. They're a very adaptive organism. They've through time and space of Earth's geologic history. So they just have, their pumping is very intentional. I would say that now there's sort of a shift to, they're not so much feeding, there's not so much pumping to feed, which they are, but also to get rid of metabolic wastes. So any living organism, we're always things and then we need to get rid of our wastes. And so they produce waste just like we do. So when they're pumping, shooting their metabolic wastes as far away from them as possible. And that's also a really great competitive advantage. So I've started to focus more a little bit on that's maybe why they're pumping, or at least equal to feeding.
Let me follow up. When the microbes are fermenting the seawater, you showed that they're producing a nitrate.
For example,
now, do the sponges themselves produce the nitrate for themselves or for the waste?
Well, so that's also a very interesting question. So you could say that they're producing the nitrate because they might need it, for the sponge might need it. So actually the sponge also hosts some photosynthesizing microbes that give it energy. So it might kind of adjust its microbial community to its needs, but also example that I gave, they're taking the that one example that I showed, they're taking up ammonia, which is a waste product, and they're getting the microbes to turn it into nitrate, which is relatively harmless to a sponge compared to ammonia. You're taking something that's very poisonous to itself to something that's less poisonous, and then you can pump it away. So I think in that case, they're actually trying to turn it into something less harmful and getting the microbes to do it. The sponge itself is just kind of like a living apartment. It's really the microbes that are doing all of it.